Now, I do want to have you look at ATP here just for one moment. So ATP is adenosine triphosphate. Adenosine, that's the same adenosine you know from nucleic acids. And triphosphate, phosphates are charged. And two phosphates together are not terribly happy. Phosphates, think of phosphates as a firecracker. When you phosphates, I mean, seriously, when you're looking at fireworks, that's phosphate right there. Poof. So adenosine, ATP, don't think of ATP as like a coin you stick in your pocket. Think of it as a firecracker that'll go off at any moment. And the moment it goes off, that energy that it releases when it goes off, that's what drives whatever process it is that it happens to be close to at that moment in time. That's how ATP functions. Um, but so ATP does it does chemical work. So it's attached to something. And the moment it fires off, it, sparks this energy. It the moment it shoots off, it changes, um, the configuration of that transport protein. And this one here, let's not talk about the mechanical. So the mechanical work here, be very careful of that. The movement does not require ATP, the release does. But that's neither here nor there. And let's talk real quick about how enzymes function. So how do enzymes help with this process of the transfer of energy? Enzymes are catalysts that decrease the activation energy needed for a reaction to begin with without being consumed by the reaction. Memorize this one. So I told you cars are kind of like us, right? Well, in terms of how they get their energy, how they utilize energy. But cars are a lot hotter than we are. So the activation energy required, the amount of heat that it requires for this reaction to work out in a car is not working for us, right? So how do enzymes help with that? I've always hated this, but I'm going to show you the chart anyway. So the way you read this is the level of energy, and this is the progress in the reaction. These are your reactants, these are your products. And what you need is you need to overcome a level of energy first in order to get this whole thing started. And then once you get it rolling, it'll, like you got to push the thing up the hill for it in order to go down. And what the enzyme does is it lowers this. It makes you not have to exert as much energy to make this happen. Now, I have forever in 3 days in my life as a student gone, how? But how? And then finally it came to me, think of enzyme as three-dimensional structures. Because that's what they are. They're three-dimensional structures that do things. And because they're three-dimensional structures, they can hold things, they can hold partners closer together. Part of why things require energy is because things don't accidentally meet unless they hit each other a lot or they have to hit each other harder. If you've got a vice grip that holds things in place, that's going to be very helpful. So let's look at this enzyme here real quick. So enzyme, when you're striking a match, the way you're striking a match onto you're rubbing it on something, right? And that creates heat. And this creation of the heat is what causes the match to strike. That gives you the energy. The heat gives you the energy to strike the phosphorus in the match and light up a match. When I teach this class in Bio 113, I usually stand there and I look at a pencil. I have a pencil in my hand. And I talk about how, what would be required to break this pencil in half. And I kind of hold it in my hand kind of as a vice grip kind of thing. Now, The other way you can light and anybody who's who's my age, who's played with matches when we were little, anybody know there's other ways to light a match, right? You don't have to necessarily strike it and you create the heat. You can bring another match close to it and you have the same effect. And that's essentially what an enzyme does. An enzyme holds on to the set of matches, the unstruck matches, and it has the light on the other side, the ATP, the little firecracker, and it basically brings together those two partners. And when it brings those partners together, then all of a sudden the reaction can happen. So it by simply a three-dimensional change of the configuration, this thing can make a reaction happen that otherwise would require a lot of energy input. So how does this happen? The way this happens is like with the enzyme, right? What's the process where the enzyme doesn't get used up? Because remember, an enzyme is a tool. It's kind of like a hammer or a vice grip. It's something that's not used up in the procedure. So you start out with the enzyme that has what's called an active site, which is the three-dimensional pocket that whatever is going to bind to it, whatever partners are going to go in there, is going to bind. And as it floats around, it just, floats around. And then it encounters the substrate that has the perfect fit that fits just perfectly into that fold. And when it does, when it fits into that fold, then the presence of the substrate because enzymes, remember, enzymes are proteins, and so proteins they're held together with like hydrogen bonds and kind of wiggly kind of structure, so they can move. Which is very important for proteins that they can do that. And so what you can imagine that this, for example, just the presence of this makes this protein, makes this enzyme move into both direction, which then exposes this bond to the water, to the environment. That gives water here a point where it can attack. And remember, water is the thing that takes everything apart and glues everything together. So the addition of water is going to take this thing apart. The moment both of those things are separate, glucose and fructose, they change configuration just enough that they don't fit anymore, and so they're released. And the enzyme goes back to its structure where it was before, to its configuration where it was before. And that's basically how enzymes function. So enzymes don't get used up. They function by bringing partners together, by holding on to something, by that kind of thing, changing configuration, that kind of thing.